Hi everybody, and welcome to chapter 27, and this is our very last lecture of the semester. So I just wanted to thank you guys for sticking with me throughout the whole semester. I know it hasn't been easy. This online format is very new to a lot of people and it's just kind of difficult. So I'm sorry I couldn't be there in person with you guys, but thank you for taking my class. I really appreciate it, and I hope you guys have learned a lot over the semester. So you have my email if you guys ever want to contact me again or anything like that. If you want to rate my professor, whatever, honestly, I don't care. I hope you guys had a really great semester, and I wish you all the best in luck in life and everything else. So hopefully everybody stayed safe, and great job on finishing a semester. I know biology is not easy for anybody, especially when we have to do it like this. So I really appreciate you guys sticking with me, and I very much appreciate it. So thank you. With our very last lecture, chapter 27, short and sweet, let's get to it, on the nervous system. So this is going to be nervous system, sensory system, and locomotion. So let's just dive right in. All right. So when we're talking about the nervous system, really what we're talking about is we're talking about our body's communication system. So this is how it's communicating. This is how we're maintaining that homeostasis that we talked about in chapter 21. Very important to keep an internal environment stable, because if it's not, like I said, all these different processes that are going on in your body are going to get disrupted, and that can lead to really bad things, including death. So not something that you want to get necessarily disrupted. So let's go ahead and look at this picture right here. I'm going to see if I can zoom this in a little bit. There we go. All right, so let's get up close and personal right here. So what we're looking at here is this is a neuron. So essentially this is one of your communication cells, sending signals from one cell to the other. And this is exactly how this happens right here. And you can follow along. So this is what's called the signal direction, meaning it's starting here and it's going somewhere else. So starting here at what's called the dendrites, and those kind of look like those little arms, those little fingers that are kind of coming out. Those are called dendrites, and those are going to be what's receiving the cell. So receiving the signal. The part of the cell that's receiving the signal, that's what I was trying to say. Okay, right here, this big part that contains the nucleus is called the cell body. So you can think of this is one of your neuron cells. And so that's kind of like the bulk of the body. So you have these little dendrites that are sticking out here just to receive a signal that goes by. And then this body right here, the nucleus, is going to almost interpret the signal and then send it off. Okay, so the signal is going to come down here from the dendrites. It's going to transfer all the way here into each one of these little right here. These are known as supporting cells. And they're essentially going to be sending the signal from one to the other, to the other, to the other, to the other, and they're connected. And they're connected via this myelin sheath. And this sheath is kind of just like a protective little coating that kind of helps to like insulate and like almost like protect the signal. Think of like wiring on the back of your TV. Those wires aren't just like sticking out, right? They're all covered in that little rubber. That rubber tubing, you can kind of think of like the myelin sheath, like it's protecting, it's covering the signal, right? That's that part of the cell that's sending the signal. So this right here is the signal pathway. Again, we're coming from here, we're going down into here, right? This whole area right here is known as the axon, also known as the nerve fiber. So this is, again, part of your nervous system. So this is what's going to be sending those signals, right? If I stick my hand in a fire, it's going to burn, right? And those cells that are here, those are receiving the signals. My little receptors, my skin receptors, are going to receive that signal and be like, there is something wrong. And then like lightning, like electricity, and it literally is. In fact, we can measure the amount of electricity going through our body when it does this. It's going to be like to our brain. It's going to send that signal. And our brain is going to send a signal right back. Okay, here's what I do when my hand is burning in a fire. Just as fast. Again, it's like electricity. It's like plugging it in. It's almost instantaneous. And then my body is going to go, oh, I'm hurting. I need to pull my hand out of the fire. So this is the whole point that we're doing this, that we have these nerve cells in this nervous system, is to communicate. Not only to communicate with what's going on inside, internally, but externally as well. So then here in our axon part of our little neuron is our signal pathway. So again, we're sending our signal through the cell. That's what I mean to say. Boop, 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 to what's called the synaptic terminal. So essentially this is one big neuron cell right here. Okay, and then what you would have over here is another neuron cell. So this synaptic terminals would actually hit up with these dendrites here of another adjacent neuron. Okay, so then the signal goes here, boop, 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 and then it would go to another one, boop, and that's how, again, what's called signal transduction. It's the transduction of the signal going through, just like electricity, one side to the other. Okay, so you can literally think of this as like communications in almost split fractions of a second, because one of them is triggering the other, which triggers the other, and it's like boom, 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 boom. And it happens really, really, really quickly, which is just awesome. Because again, how long do you want to keep your hand in that fire? Right? Not long. You need to pull that out. Otherwise, you're going to burn. You're going to hurt yourself. So we evolved to have these really amazing things like neurons to help us communicate internally and externally with our bodies to figure out what is actually going on, and therefore what kind of response we need to exhibit. Right? Remember when in chapter 21 we were talking about like what happens if I get a temperature, right? You're going to start sweating to cool down your body. Same thing if you're too cold, you're going to start shivering. These are all sent via these little neurons. We're sending signals from our, what's called the peripheral nervous system to our central nervous system, but we're going to talk about that in just a second. Now, these let's go down to each one of these little structures again that I've already kind of talked about, but we're going to go into detail. So remember those little finger-like structures on the end? Those are the ones that are picking up the signal. Those are the ones known as dendrites, right? They're receiving the messages. They're receiving the signals. Okay? The axon is where it's actually traveling down from, also called the nerve fibers, right? It's that long part with all those little cells that are talking to each other covered in that little myelin sheath. Finally, it'll reach that synaptic terminal. Again, terminal kind of just means the end. So it's reaching that end because it's going to hit an adjacent neuron cell at its dendrites and then sending the signal to the next one, which is going to go, again, one to the other, to the other, to the other. The speed, the really, really rapid speed, is also helped by that myelin sheath that we talked about. Remember that rubber coating all those little cells? It supports it and it kind of like insulates the nerves, right? We don't want our nerves firing for no reason. We don't want them sending fake signals or getting damaged by anything. Nerve damage is a big thing to worry about. So these myelin sheaths kind of help protect each one of these little neurons that we have. Now, getting to the difference between the central nervous system and the peripheral nervous system that I just mentioned is actually pretty easy. So you've heard of your central nervous system, I'm sure, because that's kind of the important one. That's your brain and your spinal cord, right? Your brain allows messages and thoughts to be interpreted and stuff like that to give actions. And your spinal cord essentially carries them out. If one of those two things don't work, then you can get things like paralysis. So that's not good. The peripheral nervous system is essentially anything branched off of that. So you have your brain and your spinal cord. That's the central nervous system, right? All the little nerves that are going down my arm to my fingertips to my toes, that's all the peripheral nervous system. So you can kind of think of those more like motor neurons, right? Those are the ones that are actually going to, like, if I want to go grab something, I'm going to think about it, and then my motor neuron, my central nervous system is going to think about it, send a signal to my peripheral nervous system, and I'm going to go grab whatever I'm going to grab and do whatever I need to do. Okay, so the central nervous system is like your control center. This is your brain. You're controlling all of your systems, right? The centralized nerve system. The peripheral nervous system, or PNS, so there's CNS and PNS, is mostly just nerves. And again, those are those nerves that are traveling down my arms to my fingertips and stuff like that. Those are going to be sending the signals to like grab stuff, or if I'm burning my hand, it's going to send it through the peripheral nervous system to the central nervous system that's going to go, cool, pull your hand out of there. And then it's going to send it back to the peripheral nervous system and it's going to go, cool, I pulled it out. So again, these basically, they're working together all the time. And this is, again, a communication system. So we want to make sure that our body's kind of talking to the rest of our body to make sure that everything is the way that it should be and everything is maintained nice and healthy and stable. Now, like I said, they do work together. So essentially what's happening in the centralized nervous system is integration, or like what's happening. So on the outside, right, is you're going to have your sensory input. This is going to be your stimulus. Okay, so in this case, we have a mosquito landing on our skin and he's going to bite us. We're going to be able to feel that. So then it's going to send a signal, right? This is a sensory neuron through a sensory signal. So like it hit one of these little receptors that we have and we say, oh, I can feel that. Right now it's sending a signal to our brain. Our brain is then going to take it, it's going to integrate it and say, okay, I received that signal, thank you. Now what do we do about that? Well, pain is not a good thing, so let's stop the pain. So then what you're going to do is through what's called an interneuron, and interneurons essentially are like neurons inside your brain. So your brain gets a signal. Well, not just this part of your brain needs to have that signal. What if, the action part of your brain is back here? So then the interneuron will send it from here to here. So it sends it inside your brain to the appropriate spot where that part of your brain will actually interpret the signal and say, what do we need to do about this? Right? If it's going to send it to your hormones and now you're producing more LSH hormones, well, that's not really going to help you, right? You need to send it to one of your motor neurons to actually be able to pull your hand out. So that's what, again, those interneurons are going to do. It's going to send a message inside your brain. So through this interneuron, we send it, okay, great. We're going to send it to our motor neurons and say, hey, motor neurons, you get moving, right? So this is going to be part of, again, both of these are the peripheral nervous system, and this right here is the central nervous system. And then through that motor input right here, output, our motor neuron, we're going to actually respond and go, ah, boom, we've killed the mosquito, we've stopped the pain. So integration, again, comes from the stimulus through the sensory input. Oh, I feel something on my arm. And your brain is going to go, okay, cool, where do we send that? Let's send it to the motor part of our brain. Through an interneuron, it sends it back here. This part of our brain goes, yeah, move your hand and go kill that thing. Great. So the signal goes back out to your arm. That's, again, the peripheral nervous system. And then your arm's going to go, and you're going to kill that mosquito. Hence, stopping the pain. Okay, so again, we're getting these sensory neurons. This shouldn't shock you. It's a sensation. This is where our senses come from. We've seen something, we smelled something, we tasted something, we felt something, right? Those are all the senses. So that would be the sensory input that we're getting. During the integration, again, that's your brain taking the signal, going, cool, cool, cool. What do I do with that? All right, send it to that department. This is the department that handles that pain, right? The pain center. So it sends a message, again, from where it receives it to the part of your brain that actually needs to handle that. So that's sent through what's called an interneuron. And then finally, your motor neurons essentially are your motor output. What do we need to do to fix this? In this case, we don't need to sweat or produce a hormone. We need to physically smack a bug that's on us. So that would, again, be the motor section of it, or the motor part of that. Okay, so the way we do this is we send it through what's called an action potential. And an action potential is the potential for action, right? So we send an electrical signal that goes, cool, we need something to happen. Okay, and until that signal gets sent, that a little electricity gets sent, nothing is going to happen. It's going to remain at what's called resting potential. Okay, so again, this is the potential. We're just resting, but it has potential, right? Remember, like kinetic energy and potential energy, the energy that it could have? Same kind of thing right here. So we're at the resting potential. We're waiting for our signal. We're waiting for something to come up and say, hey, I need to go smack that bug. I need to make something happen. Okay. So, like I said, this is going to create that action potential. The action potential is that signal, that electricity that needs to go, oh, cool. Now I can actually pick up my arm and move it and smack to actually kill the bug. So this is the physical reaction that you're going to see. So, essentially what is happening is we're kind of changing the charges internally and externally. We're just kind of messing with the electricity. And again, the positive and the negative charges are a form of conducting electricity. So that's what I mean when I say this electrical signal that gets sent. So you have your resting potential right here. This is your little inside, the inside of your neuron. You can see it's positive on the outside, negative on the inside. What happens is some of these positive ions get pushed to the inside of the neuron. What that's going to do is it's going to, and that's created by a stimulus. So the stimulus is actually going to stimulate some of these ions going inside, right? And so through these little ion channels, you're going to get, if you get enough of these little positive ions, you're going to get that electricity reaching its peak. And that's what's called when the action potential is triggered. So like nothing is going to happen until you reach a certain electrical charge. And really, we can measure this. And I did this in my physiology class, which was super cool. So essentially what you're doing is you're adding a little bit of these like positive charges, and you're like nothing, boom, and it spikes. That spike is that action potential that you finally reach the threshold and you've pushed forward. You've now created your action. So you need enough of these positive ions coming in to actually get that electrical charge enough to push your action potential forward. So as these like I said, they increase right here, the inside becomes more positive, the outside becomes a little bit more negative because again, they're kind of reversing, they're shoving their way inside. And then eventually what's going to happen is you're going to shove those positive ions back out, creating that positive outside that you had in the beginning and that negative inside that you had also. So it's kind of like this nothing, blip. And when that blip is, you're getting a reversal. You're getting the positive ions in, the negative ions out, but it's just temporary. As soon as that's gone, you go back to that resting potential. So we're going to actually watch a little video right here. So if that didn't help explain it enough, don't worry, because I'm going to come right back at you here with a quick video.
This baseball player springs into action, his nervous system rapidly processing information in a network of nerve cells or neurons. Each neuron receives input from one or more cells. In response, the neuron may generate an electrical signal known as an action potential that travels down the length of the axon. Let's take a closer look at the plasma membrane of an axon. Even without an action potential, the axon is a busy place with many ions moving across its membrane. Much of this ion movement is driven by the sodium-potassium pump. Using energy from ATP, sodium-potassium pumps actively transport sodium ions out of the cell and potassium ions in, creating an uneven distribution of charge across the membrane. Some potassium channels are open all the time, allowing potassium ions to leave the cell. As a result of these ion movements, the inside of the cell is negative relative to the outside. This condition is called the resting potential. The membrane of an axon is also packed with gated ion channels that open and close during an action potential. At resting potential, the gated channels are closed. If a stimulus changes the distribution of charge across the membrane sufficiently, the gated sodium channels open. Movement of sodium ions across the membrane makes the inside of the cell more positive. This reversal of the charge distribution causes the gated sodium channels to close and the gated potassium channels to open. As potassium ions move out of the cell, the original charge difference is re-established across the membrane, closing the gated potassium channels. This sequence of events is called an action potential. The sodium-potassium pump restores the distribution of ions back to their previous levels at resting potential. Let's look at how the action potential is conducted along the axon. As the change in charge difference across the membrane spreads from open sodium channels, other sodium channels farther along the axon begin to open. The original sodium channels close, and adjacent potassium channels open. As potassium ions move out of the cell, the original charge difference across the membrane is restored, and then the potassium channels close. Meanwhile, new sodium channels open, followed by the opening of new potassium channels and the closing of sodium channels. In this manner, the action potential is propagated along the axon of the neuron, eventually reaching another cell. The information carried by this action potential will be processed with other information, permitting this baseball player to make a spectacular catch.
We've looked at the resting potential in a neuron that is not transmitting nerve signals. What happens when the cell is stimulated? A stimulus may change the permeability of the cell membrane to sodium or potassium ions. This alters the distribution of charge in the cell body, and if the change is great enough, it triggers a signal called an action potential that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close in response to voltage changes across the membrane. When no action potentials are being transmitted, most of these channels are closed. An action potential begins when voltage-gated sodium channels open and sodium ions rush into the cell. The plasma membrane becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close, while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The plasma membrane returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in, and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. The efflux of potassium ions during the falling phase of the action potential restores the negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, re-establishing the resting distribution of sodium and potassium ions.
Let's look at another action potential as it moves along the membrane. This time we will measure the voltage changes that occur in one spot along the membrane. A slight change in polarity causes the voltage-gated sodium channels to open. Sodium ions rush in, causing a reversal in membrane polarity. Sodium channels close and potassium channels open. As potassium ions rush out, the membrane returns to being negative on the inside. The sodium-potassium pump then restores the balance of ions present at resting potential.
Okay, so I hope that really helped you, those two little videos, and you can always go back and rewatch them again. They're in your PowerPoint lecture. But I really hope that kind of gave you a visualization of what's happening. It's kind of harder to explain that the charges are changing on the outside and the inside, and then it's moving along the axon. So hopefully those videos really helped you guys. I know they helped me when I was studying. So. All right, so again, essentially an action potential is just a change, right? It's a change throughout the axon over time. So what we're looking at is a specific spot that gets changed, kind of like what she was talking about, the wave action, or even like a domino action. So you set the action potential off, and then it travels boom, 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 one affecting the other, affecting the other. And again, so you have that negative on the outside, positive on the inside, switch that you see here, and then it moves down the line until, again, you've got that reversal going back to the original positive on the outside, negative on the inside. So hopefully that would help you guys. If it didn't, luckily for you guys, we have a crash course video, your last of the semester, and I know I can't do my last lecture without having one last crash course. So let's go see our friend Hank real quick.
What if everything you did, thought, and felt could be communicated by pushing a button? It'd be like using the world's simplest app, one that just sends out a little ping, always at the same volume and length, to communicate everything from "it sure is cold in here" to "I love churros" to "boy, I sure would like to breathe sometime soon." Well, that is actually exactly how your neurons send all the impulses responsible for every one of your actions, thoughts, and emotions. When a neuron is stimulated enough, it fires an electrical impulse that zips down its axon to its neighboring neurons. But they've only got one signal that they can send, and it only transmits at one uniform strength and speed. What they can vary is the frequency or number of pulses, like this is distinct from this. And your brain can translate these signals, reading them like binary code, organizing them by location, sensation, magnitude, and importance, so that you know the difference between "turn up the thermostat" and "oh my gosh, I'm on fire." That buzz, that nerve impulse, is called the action potential. It's one of the most fundamental aspects of anatomy and physiology, and really life in general. It's happening inside of you right now, and we want to make sure that you understand what all that buzz is about. Before we delve into how neurons communicate, we gotta first understand a little bit of our old friend electricity. Basically, think of your body as a sack of batteries. No, I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positively or negatively charged than others. And because opposite charges attract, we need barriers or membranes to keep positive and negative charges separate until we're ready to use the energy that their attraction creates. In other words, we keep them separated to build potential. A battery, just sitting on its own, has both a positive and a negative end, and the potential to release energy, but it doesn't do anything until it's hooked up to a flashlight or a phone or a kid's toy that lets those charges move toward each other, on the way converting electricity into light or sound or children's laughter. In much the same way, each neuron in your body is like its own little battery with its own separated charges. It just needs an event to trigger the action that brings those charges together. If you're thinking that this sounds more like engineering than anatomy, that might not be a bad thing. It might even help to think of your neurons in the same terms an electrician might use. Voltage, for example, is the measure of potential energy generated by separated charges. It's measured in volts, but in the case of your body, we use millivolts because it's a pretty small amount. In a cell, we refer to this difference in charge as the membrane potential. The bigger the difference between the positive and negative areas, the higher the voltage and the larger the potential. And just like there's voltage in your body, there's also current, the flow of electricity from one point to another. The amount of charge in a current is related both to its voltage and its resistance. Resistance is just whatever's getting in the way of the current. Something with a high resistance is an insulator, like plastic, and something with a low resistance is a conductor, like metal. Now, when we talk about these concepts in terms of you, we're typically talking about how currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membranes. And again, these membranes separate the charges, so they're what provide the potential to convert the electricity into something useful. Okay, now that we got electricity 101 down, let's see how it works inside your nervous system. A resting neuron is like a battery just sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential, and it sits at around negative 70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. This creates a difference in the concentration of sodium and potassium, and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient, and you probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around negative 55 millivolts. But some others are ligand-gated channels. They only open up when a specific neurotransmitter like serotonin or a hormone latches onto it. Then we also have mechanically gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions. This movement of ions is the key to all electrical events in neurons, and thus is the force behind every single thing we think, do, and feel. Of course, not all of your body's electrical responses are the same, and neither are the flows of ions going in and out of your neurons. If only a few channels open and only a bit of sodium enters the cell, that causes just a little change in the membrane potential in a localized part of the cell. This is called a graded potential. But in order to send long-distance signals all the way along an axon, you need a bigger change, one big enough to trigger those voltage-gated channels. That is an action potential. And your best bet for making that happen is to depolarize that resting neuron. I mean, cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It all starts with your neurons sitting there at resting state. All of the ion channels are closed and the inner voltage is resting at negative 70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now, the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about negative 55 millivolts. Remember that number, because this is an all-or-nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. So kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, You're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about positive 40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them. And that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to negative 75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level. Now, when part of an axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter how strong. This is called the refractory period, and it's there to help prevent signals from traveling in both directions down the axon at once. So that is the surprisingly simple app that your nervous system uses to let you experience the world. And because the voltages in this process are always pretty much the same, the initial threshold around negative 55 millivolts and the peak depolarization at 40 millivolts, your neurons only communicate in a single monotone buzz. It doesn't matter if it's a spider on your knee or an elephant, a paper cut or a stab wound, the strength of that action potential is always the same. What does change is the frequency of the buzz. A weak stimulus tends to trigger less frequent action potentials, and that includes if the stimulus is coming from you, like your brain telling your muscles to perform some task. If I need to do something delicate, like pick up an egg, the signal is low frequency. But a more intense signal, like trying to crush a can, increases the frequency of those action potentials to tell your muscles to contract harder, and the message turns into something that you can't ignore. Action potentials also vary by speed or conduction velocity. They're fastest in pathways that govern things like reflexes, for example, but they're slower in places like your glands, guts, and blood vessels. And the factor that affects a neuron's transmission speed the most is whether there's a myelin sheath on its axon. Axons coated in insulating myelin conduct impulses faster than non-myelinated ones, partly because instead of just triggering one channel at a time in a chain reaction, a current can effectively leap from one gap in the myelin to the next. These little gaps are the delightfully named nodes of Ranvier, and this kind of propagation is known as saltatory conduction, from the Latin word for leaping. But what happens when an action potential hits the end of its axon and is ready to do more than leap and jump all the way to another neuron? That you will find out next time. Today, you learned how your body is kind of like a big bag of batteries, and how ion channels in your neurons regulate this electrochemistry to create an action potential from resting state to depolarization to repolarization and a brief bout of hyperpolarization. Thanks for watching, especially to all of our Subbable subscribers who make Crash Course possible for themselves and for everyone else. To find out how you can become a supporter, just go to Subbable.com. This episode was written by Kathleen Yale, the script was edited by Blake Testino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins and Michael Aranda, and our graphics team is Thought Cafe.
One more thing before you leave. We like Crash Course a lot, and we hope that you like Crash Course a lot, but I kind of feel like Crash Course is only useful for a certain segment of the population. Like, once you get to a certain age, then it's...
Alright, that was your last Crash Course video this semester, guys. Remember, he's a really great resource, so if you want to go on and subscribe, give him the credit that he's due, because he does a lot of really great work, and that whole channel does. So make sure to go at least like him or something like that. Alright, so guys, that is it. That is biology. Well, introduction to biology. I really hope you've enjoyed the course. I know it's not easy stuff. You guys have done really, really amazing work. I super appreciate it. Thank you guys for all that you've done. I hope you guys stay safe. I hope you do amazing in the future. If you ever need to contact me about anything, just go ahead and email me. Let me know. Otherwise, great job on a wonderful semester. Sorry I couldn't be with you in person, but hopefully you enjoyed your class anyway, and have a wonderful break. I hope you guys do great with finals. Good luck on everything, and yeah, take care.